Alright guys, today we're going to talk about some pretty interesting stuff. We're dealing with genetic engineering today. We're going to talk about cutting and splicing genes between different organisms. So let's dive on in here. So genetic engineering is sort of a way for us to get the desired alleles. If you remember, your genes, your DNA is the code for your genes, and when we're talking about things like, oh, this plant has big T, little t, or big T, like those are really representations. Those alleles are representations of specific genes that maybe we desire in the plant, or maybe we don't desire in the plant. So is there a way that we can edit the genome of a specific plant to insert or delete or change the actual genes that code for these alleles? And yes, there is. So we're going to talk a little bit about sort of how that's done. This technology is known as recombinant DNA technology. You'll want to know that for the test, because essentially what we're doing is we're recombining DNA from different sources, right? And remember the central dogma of biology, the whole idea, the concept that your DNA is the code, and we do transcription translation on that to make the protein. So we transcribed it into messenger RNA, and then we use ribosomes to read the messenger RNA during translation and create actual proteins that do stuff or are stuff. Everything from enzymes to like what you are. So being as it is, the code of life is the same four bases in every single thing that's alive. Everything from a worm to an insect to you and me to an apple. We all use the same type of setup and system for reading that code. So it doesn't matter which organism we're looking at. We can take genes from any organism on Earth, living or extinct, if we can get the DNA, and we can insert it into another organism, and that organism's machinery can read the DNA. And that's how universal DNA actually is. So this is known as recombinant DNA technology. We're recombining DNA from two different sources, and they could be as different as a plant and an animal, right? So let's discuss this a little bit here. The steps in order to do this require us to cut the DNA, okay? And we've already heard a little bit about restriction enzymes. Restriction enzymes, which we have whole catalogs of as biologists, where we can order the ones we specifically want. I think these were originally discovered in bacteria, and then we sort of use that technology to cut other DNA. So some of these restriction enzymes, they cut straight across the strands, and they make what's called a blunt end. Now, in order to be really useful for the most part, we want to be able to cut in kind of a zigzag pattern. In other words, remember DNA is two strands. We want part of one strand sticking out and then part of the other strand sticking out. And that leaves us these things that we refer to as sticky ends. And those sticky ends, we can then re-anneal or stick on a different piece of DNA and kind of hook the whole thing back together. So when we cut the DNA, we need to have a way of basically putting the puzzle pieces back together again. And that's why we look for things that will cut in this fashion. And EcoRI, which is an enzyme we discussed a little bit already, cuts at specific, what are known as palindromic sequences in the DNA. So a palindrome, if you remember from English class, is a sequence that's the same forwards as it is backwards. So let's take a look at what EcoRI does, and it'll make a little bit more sense. So here's a double strand of DNA, right? Just like you're used to. It reads off TAGAA, TTCGGA, and you've got the corresponding bases that would pair with that across the bottom. Okay? There's a section in here that's a palindrome, and it's a palindrome where EcoRI specifically cuts at. Okay? If we look right here, here is our palindromic sequence, GAATTC. Okay, that's the sequence that EcoRI as an enzyme looks for to make a cut. And here's how it's going to cut. It's going to go ahead and basically chop that DNA like this. So you can see that once it's cut, part of the bottom strand is going to be sticking out, and part of the top strand is going to be sticking out. Then if we want to do some engineering, all we have to do is have a similar cut on the DNA that we want to insert that has the same corresponding base pairs sticking out the ends of it, and we can very easily insert it on top of that, and it's going to want to pair. As long as our new strand, for instance, read AATT, and then whatever we wanted after that, we could just stick that right on there because the AATT would match with the TTAA, right? So when that thing is cut, we might see something like this, and now we've got two sticky ends. So that's kind of step one to any sort of genetic manipulation, any sort of genetic engineering that's going to be done. You've got to get the right enzyme to cut with, and it needs to produce some sticky ends that we know, and then we need to engineer some DNA that's going to fit onto those sticky ends. Okay, so let's say we've engineered our DNA, we figured out how to cut the DNA we want to insert it into. The next step is to find a way to get it inside. And there's different methods of doing that, and I listed 3 of them here. And you've probably heard a little bit more about this in the news because we're using some of this sort of a technique in some of our new vaccines even. So let's take a look. The first one here I've got listed is to use a virus as a host. So what we could do is we could take the DNA that we want to put into the cells that we care about and insert it into a virus. Ooh, that's interesting. So what we need to do to accomplish that is we need to make sure, first of all, that the virus is broken in such a way that the DNA is deactivated that's viral DNA. So we need to probably remove a bunch of it. So this is tricky stuff. Then we need to get the DNA that we've created, engineered, or taken from another organism, inserted into that virus. Okay, next step is we need to get that virus. Basically, we're going to allow that virus to infect the host cells that we care about. This technique is really similar to some of these vaccines you see coming out, like the Johnson & Johnson vaccine, the AstraZeneca vaccine that's coming out for COVID, because what they're using is a virus that's actually a chimpanzee cold virus, okay, and it will infect human cells. And so what we did was we basically gutted this virus, inserted the code that we cared about putting in there, infect your cells with it, and then it's going to, the virus is the deployment system. It will match up with your cells, proteins on the surface, and then it will inject the genetic code into your cells. And then your cells do what your cells do. They read the code, they make the proteins. In this case, we're putting a spike protein code from the COVID virus into a different virus, so your cells are going to make spike proteins without ever actually making any active virus that would harm you. Then your cells are going to present those spike proteins to your other cells. Your cells will have an immune response, so they'll start to recognize what those spikes look like. And then, of course, if you get infected with the real thing, the real deal, the real COVID, then your body already knows what it looks like and can mount a quick immune response and knock it right out. So that's probably going to be the future of vaccines to come. So it's a really exciting thing. Let's look at the other 2 I've got up here. These are pretty common. All 3 of these are fairly common methods of engineering. So plasmids. What a plasmid is a little ring. It's a little tiny ring of DNA, and bacteria have these, and so that's where we got this idea. So bacteria have they don't have DNA that's stored in a nucleus. It's kind of all strung out inside of the cell, and along with that, they have these tiny loops of DNA that are special to them, because what they can do is they can pass these plasmids between other bacteria. So bacteria are weird things. First of all, they're ancient. They've been around forever on our planet. So they've got some unique tricks for dealing with DNA. And one of those is to make a plasmid where they can actually share code with another bacteria. And we've mentioned that a little bit a few units ago. We talked about how bacteria can do this thing where they actually hand off some of that DNA with the conjugation bridge to another bacteria. And then that bacteria suddenly can read that code and make itself, say, immune to an antibiotic. So this is one way bacteria can sort of adapt to their environment. Anyway, what we want to do is we take a plasmid, and it's a little ring of DNA, and we cut it. We get some sticky ends, we insert the DNA we care about into that plasmid, and then we give it to a bacteria. And the bacteria is going to take that up. So it's going to read it, it's going to produce whatever our new code is on there. And we have been using this technique to make a lot of stuff. So bacteria are tremendously useful for genetic engineering. They're like little factories, and they really easily grab DNA and just kind of read it off. Another method is known as micropipetting. So we could, if we have a larger cell we're dealing with, we could actually just squirt the DNA we care about directly into the cell. So that's another method as well. There's a lot of ways to do this, but remember, kind of the key takeaway from a lot of this is that the code is universal, right? It doesn't matter like where the code came from, whether we make it, whether a virus made it, whether it's some mutation, some new variant somewhere, whether it came from a totally different organism from a completely different kingdom of life. Every cell can read it. That is how closely related all life on our planet actually is. So here are the 3 steps again, just so you can see them in case you have them on the test. Recombinant DNA involves first finding and isolating the DNA fragment that we care about. Okay? Then we need to attach that. So we've cut out a section of DNA that codes for something. It's a specific gene that, say, codes for insulin. We cut it out, and then we want to attach the DNA to some sort of a vector. We need a delivery system. We put it in a plasmid maybe, okay? And then we've got a way to transfer that DNA fragment into the host. Maybe the viral vector does it itself. Maybe we have a micropipette and we put that plasmid right into the bacteria, and then the host cell starts to read it and make the proteins that we care about. Now, obviously, there's more complexity than this, right? There's lots of issues, and it's not as simple. We need to make sure that the code gets inserted into a place that it's not going to cause problems with the host organism. We need to make sure that the code doesn't get scrambled or anything on its way in. There's a lot of little pieces that kind of have to be worked out. It's not just quite as simple as I'm making it sound here. So once we're done with this, once we've recombined DNA into a new organism, we refer to that organism as a transgenic organism. So a transgenic organism is one that contains foreign recombinant, right, DNA in its cells. So we've completely created basically a new genome for this organism. Now, one way that we've started doing this very, very rapidly is a process that I'm sure you've heard about because it's been in the news so much, and it's known as CRISPR. And CRISPR is a really unique method, and I want to talk about it just a little bit, but I've got a short video here for you that will explain it a little bit better. You'll see some visuals. So, turns out bacteria have an enzyme, a special enzyme that can actually edit DNA, and it's known as the Cas9 enzyme. So what this thing does is it can, for a bacteria, what it does is that a bacteria can basically identify certain viruses that have attacked its cell, and it has this defense mechanism where it can once it finds that code that the virus has, it can make a copy of it as an RNA guide. Okay, and that RNA guide will match any DNA sequence that is its complementary one. So once this thing has this guide, it inserts this guide into the Cas9 enzyme, and then let's say a virus, the same type of virus, infects the cell again. It inserts its DNA into the bacterial cell. It's about to wreak havoc on it. Well, the Cas9 enzyme finds that new DNA that's inside the cell, and it reads through the whole thing. It like checks it, error checks it. And if it finds a match to its guide RNA, it'll chop that thing in half, it effectively disabling the foreign DNA that might have messed up the bacterial cell. It's a heck of a situation. But you can see that how when scientists discovered this, they were like, oh my goodness, like we could use this technique to actually edit DNA on our own, because Cas9 will take virtually any guide RNA you want to stick in there. And we're really good at making RNA. It's like something we can easily do, and you're seeing that happen with these RNA vaccines that are coming out right now. We can custom design RNA, find a way to insert it into your cells so that your body can create these viral spike proteins so we can protect ourselves against them. It's exactly what we're doing. So, Cas9, very useful for bacteria. Humans have hijacked this method now, and we're using it to do very rapid genetic engineering. And one thing that we've done, I've got a little note up here, Arctic apples. You may have heard of these. They're unique. They are non-browning apples. So you can cut them, you can bruise them, they won't turn brown. How about that? And you might ask, well, why would we care about doing that? Well, here's the deal. Like about almost half of the United States produce that we make, that we farm, gets discarded because people find it unappealing. It's not bad, you could still eat it, but who wants to eat a brown apple, right? It looks bad. So what scientists did was they recognized, they found that there were specific genes in apples that create an enzyme that causes this browning thing to occur when it gets exposed to oxygen. That's when you cut the apple or you bruise it. So some apples they found had lower amounts of this enzyme and didn't bruise as bad. Like we'd known this for a while, like some apples don't bruise as bad as other apples. And so they identified the enzyme and they selected an apple. In this case, it happened to be a golden delicious apple because it was already one that didn't bruise as badly, and they found the gene in it, and they basically deleted out this gene. They broke this gene. So it doesn't create the enzyme that would cause the apple to bruise when it gets cut, which is really kind of interesting and exciting because these things last so long, and people can still eat them. They don't taste any different. They're exactly the same as a regular apple minus one tiny little segment of DNA. So you can imagine people are freaked out by this. People are freaked out by genetic engineering. But I'm here to tell you that this is going to be the future. We're going to see this more and more and more. And we probably just better come to grips with the fact that some of our food is going to be genetically engineered. And in many cases, it's going to be better for us because it's genetically engineered. Now, I'm not saying we don't need to be careful. We need to be very careful that we're not editing something that could cause a problem. But being as it's food and your body is going to digest it, there is a little less concern for what we're doing to the actual genes that are in the food. But we need to test things and make sure that they're safe. But we also need to not be overreactionary just because we hear the words genetically engineered apple. In the future, I guarantee you, people are going to fight over whether or not they can get a specifically genetically engineered thing to eat because it's going to be better for you. We've already got plans and methods in the works for doing things like creating potatoes that lack the gene that gives you that can cause cancer sometimes. That causes a predisposition to cancer when you fry potatoes. Fried potatoes, we all know, are not great for you, but we all love French fries. What if you could make the French fry safer to eat, right? What if you could make bacon safer to eat? So those are all things that people are actually actively working on, but it's going to be a big PR thing to try and get people to understand that we've actually done something good here. We've created foods that are safer, foods that can be grown in conditions that are harsher, like for people who live in desert climates or drought climates where they can't grow things, we can create food that can survive that and can feed them. So this is a real serious thing, and I think we need to take a real balanced approach to it. We need to be careful about just suddenly discarding something because scientists have had their fingers in on it. So anyway, that's the CRISPR technique. Here's a short little video to kind of show you how that sort of plays out.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR Cas9. CRISPR is short for clustered regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then, they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA-cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care, or even cure lifelong inherited diseases.
So that's the idea behind CRISPR, and it's really a wonderful, amazing technology. I mean, it's probably one of the most exciting things that's happened, honestly, in my lifetime. So, what does this kind of look like in a sort of a really quick drawing here? We've got a DNA plasmid, which is in yellow right here. We've got a section of DNA that we want to insert into it, and what we're going to do is we're going to take and cut that plasmid. Okay, we utilize enzymes, specific enzymes that target, a palindrome sequence. We cut it, and then we insert our DNA. Once the DNA is then inserted, what we're going to do is we need to find ourselves a cell to get that into. Okay, and whatever technique we want to use to get it in there, we've got this bacterial cell. We insert this plasmid, which has the new DNA inside of it, and we have just created a transgenic organism. So now, all we need to do if we want more of it is to let the bacterial cell do what they do. Let them undergo binary fission and reproduce. And in very short order, you're going to end up with an awful lot of bacteria. But look, every single new bacteria has that same exact gene, so we've just genetically engineered an entire population of bacteria. You might be asking, what's the purpose of this? How is this useful? Well, let me get to that. But we actually can do this right here at East High School. Here are some plates with some bacteria on them and some students in the lab. And what they've done is they have taken the gene out of a jellyfish, which will glow under ultraviolet light, and inserted that gene into bacteria. When they grew these bacteria up in little colonies, and those aren't individual bacteria, those are whole colonies of bacteria that you see on the plates, put them under an ultraviolet light, the bacteria glow in the dark. Now, why would you want to make glow-in-the-dark bacteria? Well, it's cool, but is it useful? Yes, it actually is really useful. So this is oftentimes used as a tagging mechanism. So let's say a scientist wants to insert a gene for something that will actually be active, that will be an important, useful gene. Okay? How do they know that the gene actually got into the bacteria? especially it's from an organism that it's not going to provide any characteristics you're going to see in the bacteria. So you add in along with this gene a copy of this gene from the glow-in-the-dark jellyfish, and then you locate the bacteria that have actually picked up the gene, because they're not going to all grab it. So then once you do that, you can isolate the actual bacteria that have your gene in there with them, the one that you care about, because they're going to glow in the dark because they also picked up the jellyfish gene. So it's actually more than just sort of just some smoke and mirrors sort of fun thing to do. It has a real purpose. Now, why would you want to do that in kind of the end game? So one of the big reasons we want to do this is known as gene therapy. So the idea is that we can take and insert normal genes into human cells to replace defective ones. Many disorders that humans have are caused by incorrectly folded proteins or missing proteins. Proteins are enzymes, they're hormones, they're everything that does things in our bodies. So if you've got a defective one, is there a way to edit the DNA that's behind that and fix it? Yes, there is. And this is on the forefront of technology here around the world and in the United States. So the other thing that I put up here is the idea of protein therapy. So let me kind of describe that as well here. So you could say, not replace the actual gene, but can we just give you the protein that you need? We've been doing this for a long time. Insulin is a very famous example, so you might know some diabetics. If you're a diabetic, you don't produce insulin, and insulin is a very essential protein in your body. So what we can do is we can inject insulin into you, and many diabetics give themselves insulin shots. Well, originally my grandfather had diabetes, and originally he used to get his insulin. They would extract it from horses. Basically, it was kind of a horrible process, but you can imagine horse insulin is not going to be shaped exactly like human insulin. So he said it never made you feel very well when I talked to him about it. And he was alive. He's no longer alive right now, but he was alive during the time when we started doing protein therapy. So where we started actually figuring out how to give humans human insulin. And the way we did that was we took a bacteria, we inserted the human gene for insulin into bacteria via a plasmid, and then had the bacteria produce human insulin. Then we just, harvested the bacteria, took the insulin, purified it a bit, and now when my grandfather would inject himself with insulin, he was injecting actual human insulin, which was so much better for him. And he said it made him feel a whole lot better. It was a big difference. So nowadays, all insulin that you get, if you're a diabetic, comes from genetically engineered bacteria that are making it for us. We no longer have to do these things to horses. It's just right from human genes that bacteria make for us. So that's like a kind of a protein therapy sort of an idea. We give you the protein you're missing. What about actual gene therapy? So can we replace the genes? Yeah, we are actually doing this. In the United States, it's only available in clinical trials. There's no wide-scale approved uses yet. The main things we're focused on are things like cancer, cystic fibrosis, heart disease, diabetes, obviously, hemophilia, and even AIDS, HIV. There might be ways to treat diseases this way that are from a virus. How about that? So what we do is we can actually use a vector like a virus or something like that to deliver this package to basically edit your DNA. And if we do that and we do it right, this could be the future. We could fix a lot of problems. Now, this does not come without its risks. There are big risks with big rewards, right? So if we can do this properly, the rewards are going to be huge. The risks, well, let's kind of talk about them a little bit. When you introduce something new into your body, you always have the risk of sort of mounting an immune system reaction against it. And that's part of the thing that we've been watching out for very carefully with the COVID vaccines. So it can cause, inflammation. In severe cases, you can have even organ failure if your body mounts an immune response to this newly engineered stuff or to whatever we've put into your body. So that's something we need to be careful with. Another problem is that we need to target the right cells. If we're trying to deal with blood, we need to deal with the cells that are manufacturing your blood. We don't want that to, attack your liver or something like that. So we're using, say, viruses to deliver these payloads into the cell. We need to make sure that since viruses can attack more than one type of cells, that we've altered them so that they really only affect the cells that we want to target. So targeting is a problem with a lot of these techniques because you're going to hit more cells than what you want to hit with the new code. So the other issues, if you're using something like a virus as your vector here, then it's also possible that the virus could recover its original ability to cause disease because mutations can and do happen, right? So we need to make sure that we've adjusted this virus in such a way that it's only adjusting what we want it to adjust, but it's not itself recovering the ability to make you sick, because that kind of defeats the purpose, right? And then, of course, there's other issues too. Anytime you go editing genes, it's kind of, in a way, similar to mutating DNA, right? If you insert those genes into the wrong spot in your DNA, then there's a chance that that insertion could lead to the formation of tumors and cancerous things. So that's also a problem with this technique. So there's a lot of pieces to sort of work out here, but really gene therapy, that's the future. We're going to have situations where you've got some certain cancer, and we don't have to do things like chemotherapy and cutting out the tumors and things like that. We will just give you a basically an edit on your DNA and fix it. And other things like say HIV and things like that are diseases that are caused by viruses themselves. Well, the cells that are infected, one of the problems is that we oftentimes can't show our immune system that those cells are damaged and problematic. So how do we tag them? Same thing with cancer. How do we make those cells known to the immune system so that our bodies take care of it on their own? Well, genetic engineering offers the chance to modify those cells that are causing the problem, and then your body's immune response itself can go destroy those cells. So there's a lot of possibility here. It's super duper exciting stuff. Yeah, we need to be careful with it, but don't be afraid of it to the point where you neglect all of the modern technology that we're developing here. So anyway, that is gene therapy and genetic editing. There you go.